chapter, uh, the major themes in microbiology, um, giving you kind of a broad overview of the different topics that are covered in a typical microbiology course. All right, and so we were talking about the utility of microorganisms and biotechnology, genetic engineering, bioremediation, um, and then where we went from there. And then the discussion turns to the negative side of things. Okay, so um, while microbes are essential to our existence, I can't stress that enough. This is a microbial world, and without them, especially the prokaryotic and eukaryotic microbes, without them, um, life as we know it wouldn't be possible. Okay? So just know that this milieu that we occupy, that is hospitable to us, that allows us to persist, allows us to live. Much of that is a result of microbial activity. The oxygen that we're breathing is a product of photosynthesis. You've got blue-green algae in the astronomically huge numbers. I can't even begin to tell you how big of a number it would be if we were to count all of them. But the blue-green algae in the world's oceans and freshwater bodies of water. The other photosynthetic eukaryotic algae, all of these are cranking out just unbelievable amounts of oxygen, rivaling major forests with regard to their production of oxygen. So without just those photosynthetic microbes, we probably would be out of luck. I've mentioned the nitrogen-fixing bacteria. If we didn't have those, the nitrogen-fixing bacteria and protistan organisms, there's some eukaryotes that fix nitrogen. Without them, it's likely an impossibility that living things would be able to acquire the nitrogen that they need to make nitrogen-containing organic molecules like amino acids, nucleic acids, and others. So that's huge. And you've probably never even heard of Azotobacter or Rhizobium or Nostoc or any of these other kinds of microbes that are doing this photosynthesis. And then the eukaryotic photosynthetic organisms, the plants, everything from mosses on up to the largest flowering plants, they have in their cells chloroplasts. Well, where did those organelles come from? Those photosynthetic organelles? Well, their origin is bacterial. Chloroplasts are derived from gram-negative bacterial ancestors, and they now reside entirely within photosynthetic organism cells. Likewise for mitochondria. Mitochondria are the organelles that all eukaryotes have, plants, fungi, animals, and protistans. They have them in their cells, and these are the sites where we have things like the Krebs cycle happening and the electron transport chain happening. And ultimately that leads to tremendous production of ATP, which is the currency required for your cells to operate. So thank microbes. And, we've got decomposition, and then we have, antibiotic production and bioremediation and biotechnology. All of those things and many more are important, positive, beneficial attributes of these organisms. Now, there's the downside. Microbes cause disease, infectious disease. All right. And the forms that do that are known as pathogens. Now, not all pathogens are created equal. Some forms, I call them forms, species. Some species are what we call strict pathogens. So pathogens can be strict, And then there are some that are opportunistic. Opportunistic. Maybe one day I'll get good at writing with my finger. But for now, that's the best I can do. I wish there was some kind of stylus I could purchase for this. Maybe I'll look into that. But anyway, a strict pathogen is an organism that anytime it finds an appropriate host, it will cause infection, and that ultimately could result in disease symptoms. We know now, it's fairly common knowledge now that there are organisms that cause infection that are don't produce symptoms. We can have asymptomatic infections. We're learning that now as a result of the pandemic that we're enduring with the coronavirus, the SARS coronavirus 2. But that's true of many, many other pathogens. But if it's a strict pathogen, yes, it will cause infection, and very likely will cause a person to exhibit some untoward, symptoms of illness. Opportunistic pathogens are forms that normally don't cause disease. They don't cause infection. They can occur in the body and just be hitchhikers. They can just be there hanging around, doing neither good nor bad. The individual that has them, that has acquired those organisms, is said to be the host. And most of the time, those forms don't really do much to the host. By the way, that relationship, when we have an organism that lives on a host but doesn't cause them harm or cause any benefit either, that's called commensalism. Okay, just so you know, that's a type of relationship. That's a type of symbiosis. Okay, it's a plus for the commensal organism, the bacterium or the protista or whatever it might be, and for the host, it's a zero. They're not being affected one way or another. So most of the time, the forms that we call opportunistic pathogens are like this. They're usually commensal. They live on and in us, and we're none the worse for wear because of it. Occasionally, though, if that person has a compromised immune system, for whatever reason, stress, or they have another infection that they're fighting, or they're enduring chemotherapy or something such as this, that bug, as we call them commonly, that virus, that bacterium, that protistan, can all of a sudden turn on that individual and cause them to be sick. And so there's a bacterium known as Clostridium difficile. It's often just shorthanded. People refer to it as C. diff. If you're in the biomedical field, you're a nurse or doctor or what have you, you'll likely have encountered this. About C. diff. It's a very common organism that it's a bacterium, and it occurs in hospital settings very frequently. And people sometimes go in for procedures and they acquire it there, or sometimes a person endures a really powerful antibiotic treatment for an infection that they have. And what it does is it kills many of the organisms in their gut. Those same organisms that have been killed formerly were competing with C. diff, which was held in check by those organisms. So whereas a little bit of the Clostridium difficile in the gut, but not enough to make you sick. But after treatment with a powerful antibiotic, what happens is Clostridium difficile sometimes is released from that inhibition and becomes really very abundant and can cause illness then. So that's a good example of an opportunistic pathogen, one that normally is there, but just you don't even know it, and then something happens and enables it to just explode and cause major problems. Now, of the organisms out there that have been studied with regard to their pathogenicity, there's only around 2,000 different microbes out of thousands upon thousands upon thousands that are known to cause disease. So that, should be, even though right now we're dealing with a virus that's wreaking havoc, it makes it seem as all microbes are bad. The fact that there's only about 2,000 total microbes out there that cause disease, as far as we know, should give you a little bit of hope that not everything is out to get you. That's a relatively small number of things, and there are some entire groups that are not known to cause disease at all ever. Right, the archaea, this domain archaea, these organisms that have simple genomes, much like the bacteria. We'll talk about them more later. Those strange, bacterial-like organisms that occur in out-of-the-way places, those archaea, are not known to cause any disease at all. So that's pretty interesting. So anyway, there's that. Now, as far as the number of infections every year resulting from acquisition of these microbes, the number is around 10 billion worldwide, and that number probably is higher now since the time that this particular figure was first published. A new edition of the book has come out. Our world's population has gotten bigger, so more people, the more likelihood of infections. I think we're somewhere around 7 billion people worldwide, pushing toward 8 billion. And so that number 10 billion reflects the fact that when people get infected, commonly a person is going to be infected more than once annually. So you might get a viral infection at some point in the year, and then later on get a bacterial infection. Multiple infections. Many individuals are susceptible to those, or succumb to those every year. And then, as far as death due to infection, you can see the number of individuals dying due to infections is very much smaller than the 10 billion. So 13 million deaths from infections per year worldwide. And over here in this table 1.2, it lists out what currently are the common infectious agents, the bugs, if you will, the viruses and the bacteria and the protists and the fungi that cause infection. And you can see that the biggest proportion of infectious agents are actually animals, very tiny animals, helminths, as they're called. That word is helminths. Write that here. That's a catch-all word for parasitic worms. There's several different classes of flatworms in particular, but also roundworms that cause infection, and especially in places where sanitation is not fully developed, and water supplies might be contaminated and so forth. People commonly harbor these worms in their guts and in their lymphatic systems and elsewhere. And then we have parasitic protozoan infections. They're very common. All of those are horrible. If you know anything about any of those, leishmaniasis is sometimes known as a white leprosy, I think is what it's called. Leprosy is actually caused by a bacterium, but leishmaniasis is caused by a little protozoan that is vectored by biting flies, and it's just absolutely devastating to the people that acquire that infection. Trypanosomiasis is caused by little flagellated protozoa that infect the blood and destroy red blood cells, and they cause anemia and all kinds of bad symptoms from there. And of course, malaria and dysentery are fairly well known. But those things are not common in our part of the world. They're more common in less developed countries, specifically in tropical regions. And so, as far as we're concerned, they're really not a problem for us. But you can see over here, worldwide, more than 500 million, almost half a billion people worldwide will be infected with these things. Lots of very striking numbers here, and things that people frequently just are not at all aware of because of where we happen to live. We live in a place where conditions are such that we don't encounter many of those organisms. So at the top, you got diseases that can be caused by multiple different pathogens. At the bottom, we have diseases that are caused by single pathogens. And amazingly, this organism, tuberculosis, is still incredibly prevalent. It arguably is the most dangerous organism ever. That's a bacterium that causes this, Mycobacterium tuberculosis. There's different variations of it, different stereotypes, I guess you could say. But through history, it's been just a horrible killer, and it continues to infect large numbers of people, despite the fact that we have therapies and things to treat it. It hangs on and has evolved antibiotic resistance, unfortunately. So it's still out there, still a threat that looms. And you can see some of these others, some of which we have vaccines for, like for measles. And of course, we have these influenza vaccines that we have to update periodically because of the rapid evolution of those influenza viruses. But look at the numbers, numbers of cases. All right, so there are some forms of microbes that are pathogenic and can do considerable harm to people that acquire them. Anyway, so that's another aspect of microbes that has to be considered. And if you look at the top causes of death in the United States and in the world, you can see that the blue bars in each of these little clusters of bars in this histogram plot, that color there, these bars correspond to the number of deaths measured in the thousands. In each of 4 different categories. So low income countries versus lower middle income countries, upper middle income countries, high income countries. We happen to live in a high income country. And you can see that in both of these clusters of bars, the prevalence of infectious disease is it's not negligible, but it's not the most important problem. The most important problem would be chronic diseases like heart disease and metabolic issues resulting from autoimmune conditions and so on. Those are what kill people in our part of the world. In less developed countries, you can see communicable diseases. That is, infectious diseases that can be transmitted from person to person to person. Those are astronomically more important in those places than they are elsewhere. That's a pretty stark figure there that's hard to dispute. Now, as far as diseases are concerned, they're classifiable in a variety of ways. We have some diseases that are considered to be emerging. We have things that have never been seen before, novel, right? Novel infections, like the novel coronavirus that's causing this disease, COVID-19, which, by the way, is called COVID-19 because it's a coronavirus. It's a disease caused by coronavirus, and it reared its head in November 2019. This is an acronym, but severe acute respiratory syndrome coronavirus 2. That's an emerging disease. The COVID-19 that's caused by this is an emerging disease. Others that heretofore, if we go way back in history, there was no evidence of them. HIV is one of those. So sometime in the later 1970s, early 1980s is when it really took off. If you'd been alive in the 40s, 50s, 60s, and before that, there was nothing like HIV that was known. So there's that one, West Nile virus, various strains of influenza virus. We can consider those emerging. Things like Ebola, virus. A lot of viruses. These things evolve very rapidly. There are no proofreading mechanisms that are available as their genomes are replicated. Mutations happen, and it alters how the genes are constituted, and when they're expressed, it leads to changes in how those viruses affect the people that acquire them. And so new strains of viruses in particular are showing up all the time, although there are bacteria too that undergo these changes too, and sometimes become emerging diseases. In nearly all cases, these emerging diseases arise in non-human animal populations, right? So bats have been a reservoir for a number of things. Birds, the West Nile virus is harbored or has been harbored by birds, specifically those in the family Corvidae, which includes crows and other blackbirds. Chickens have been a reservoir for the SARS virus and other SARS viruses, other coronaviruses, and influenza viruses. So yeah, these a lot of diseases have their origin in non-human populations, and then when we live in close quarters with those animals, they sometimes evolve the ability to jump from one host to another. And so those are called zoonoses. The root word ZOO refers to animals. So a zoonosis is an infection that normally would have occurred in some other animal, say a bird, like a parrot. And then for whatever reason, humans began to acquire it. So that's where these emerging diseases come from. And, it stands to reason that if you have dense human populations that are commonly coming into contact with non-human animals, the virus jumps from an infected animal to a human. Very dense populations where there are a lot of people crammed into fairly small areas are populations wherein those viruses spread very rapidly. So something that's highly contagious, it stands to reason, is going to make its way through such a population rapidly. Unfortunately, our human population is bigger than it's ever been, and everywhere it's denser than it's ever been. And so we can expect these things to happen. There's really no two ways about that. So it's something that we need to be prepared for. And of course, this most recent event really emphasizes that. Of course, then there are reemerging diseases. These are diseases that historically were very significant. And then, technologies were developed to keep them in check, drug therapies and so forth. And they seem to work. But then, these are organisms and their populations are turning over and they evolve. And sometimes they find ways to overcome those inhibitions. And so they reemerge. And of course, tuberculosis, which we saw earlier, affects billions of people worldwide even now, historically was much more abundant. And various kinds of influenza. We had the 1918 flu epidemic, the Spanish flu epidemic, well, that same type of flu has reemerged in recent years. And likewise with malaria and so forth. So just because a disease had been, held in check for whatever reason, if it was never eliminated entirely, there's a chance that it could make its way back into the population. As far as I know, I think the only infectious agent that's been eliminated entirely is the smallpox virus. It still is found in some laboratories, and there has been discussion about working with that again. I don't know why anybody would choose to do that. But for now, it's not out there circulating and has been eliminated. But we'll see what the future holds. Yeah. All right, so there's that. With regard to the different things about microbes, and I could go on and on and on. And there may be some of those things that I pull back out to expound on as I think of things that I might add to what I've already said. But for now, we're going to go ahead and leave that behind, and we're going to move on and start talking about history a little bit. Okay? As I said, the reason that history is important for a scientist is so that, well, there's a lot of reasons, but the major reason is what's been done before so that you don't repeat it, right? You don't want to spin your wheels doing something that somebody has already thoroughly investigated. If somebody's put forward a particular hypothesis and they tested it and it was rejected, and if other people tested it and rejected it, it's a good bet that if you were to try to reevaluate that hypothesis, you probably would your work would lead to its rejection as well. So if you know where we've been, the places that are likely going to bear fruit if you go in those directions because they've not been done before. So anyway, that's kind of why this historical stuff is valuable. And it also is sometimes inspirational. People like to read biographies of important figures from the past to learn about what makes them tick, and there's some of that here too. People can be inspired by some of these folks. And when you walk out of a microbiology class, if you don't know who Louis Pasteur or Robert Koch, some other folks, if you don't know those names, that's not a good thing. So I would feel really, really horrible if you took my class and you didn't know those people. So I'm going to spend some time on some of these folks. So microbiology dates back about 300 years, back to about the late 1600s, somewhere in there. Really took off in earnest in the 1800s. But there were people who were doing microbiological investigations of a sort before that in the 1700s. And science was, biology in particular, was sort of in its infancy. People who were practicing doctors were, the first important biomedical scientists. They still relied on, old folk kinds of beliefs and so forth, and some of what they did. But ultimately, more and more people started catching on to the scientific method and how that worked. And it ultimately became, the way things that were done. You apply the scientific method. And we'll talk a little bit about what that entails. It's probably something that you've had before in previous science classes, but we'll revisit that here again. So some of the early things that happened, this one here is big. Microscopy. the development of microscopes, the development of polished round lenses, um, you know, little sort of convex surfaced pieces of very clear glass or crystal um, were used to alter the way light rays passed from an object back into somebody's eye and thereby magnified them and also gave high resolution clarity of the image as well. That's what we do with microscopes. We make things look bigger and we make them look clearer so we can see their fine detail. And the first microscopes obviously were very simple, but a person who sort of had a hobby that involved microscopy, making microscopes and using them, was this guy, an Englishman named Robert Hooke, who, looking at a piece of cork, which is, from an oak tree, it's part of the bark of an oak tree. It's all these little rectangular things arranged in there, stacked on each other, and saw that there was this kind of regular kind of structure that to the naked eye was not visible, and a bunch of little rectangles. And he surmised that each one of those was kind of like the fundamental building block out of which the whole was made. And he called each one of those a separate cell. And he coined that term. Now, thereafter, Hooke really didn't do much microscopy. He went on and did other things. But another important microscopist we'll be talking about soon was active in Holland, and his work was really significant. And he's kind of the first major player in the field of microbiology. We'll be talking about him. And then later there was the recognition that microbes cause disease, so this whole field of medical microbiology emerged. And then, of course, techniques for rearing microbes like bacteria and viruses in the lab so that they can be studied. Techniques that would lead to facilitation of identification of microbes and learning about their biology and so forth. All of those techniques were developed in the 19th century primarily, which is regarded as still kind of early in the history of microbiology. So one of the ideas that prevailed early on among very early microbiologists and other folks, scientists and others, was this notion of spontaneous generation, also sometimes called the theory of abiogenesis, which holds that living things can arise from nothing. This is essentially what it means, that when new organisms are found in a particular setting, they could have arisen simply by changes in chemicals that occur there. Some other kind of parent material would give rise to new organisms. Like, say, for example, if you set out a piece of meat and then forget about it for a day or two and then come back and see some maggots growing on it, if you didn't know about the life history of flies, and if you didn't see the flies actually landing on the meat and depositing eggs, you might be led to believe that the meat just simply started growing these maggots, these fly larvae, spontaneously. That some sort of vital factor occurred in the meat that enabled that to happen. Of course, now we know that idea is very, very wrong. But there was a time in history when people didn't have enough observations, weren't aware enough to be able to put two and two together and say, oh, flies did this. They thought it was spontaneous generation. So the person that dismantled that idea was Francesco Redi, I believe is how you pronounce his name, an Italian physician. He did a little simple experiment where he laid the meat out, but this time he covered it with some mesh to keep outside agents, namely flies, from getting to it, and that prevented the maggots. So it's not something that's intrinsic to the meat that's causing this. There's, life, when maggots arise in there, they have to come from other life forms. So that idea, eventually became central in microbiology with regard to the study of bacteria and others. When you see bacteria growing in some liquid broth, for example, when they start to flourish there, you can't say that they just started growing de novo from nothing, that they arose out of nothing. There had to be some contaminating bacteria present there to begin with. That was an idea that was really strongly proposed and well supported by Louis Pasteur, and then other microbiologists followed suit later. So that idea was dismantled fairly early on. So yeah, spontaneous generation does not happen. Instead, organisms, when they arise, have to come from other organisms. That's called, appropriately, biogenesis, living things giving rise to other living things. All right, so that's enough of that. That was an early major sort of milestone in the development of microbiology. It's just that realization that these microbes are coming from other microbes. They didn't just show up out of the thin air. The first person really to see microbes was this microscopist. I mentioned Robert Hooke, but the father of microscopy, really, the first sort of rock star in microbiology, was this guy, Antony van Leeuwenhoek, who was from Leiden, Holland. He's from Holland. He was Dutch. And the interesting thing about him was that microscopy, that biology, those were hobbies for him. His profession was actually buying and selling cloth. But at the time, there in the latter 1600s, early 1700s, that sort of coincided with a period when Western Europe, Britain and England and Germany and France and Holland, folks from those places were going on voyages around the world and finding new things and charting new territory and bringing back goods to sell. And among those would be things like silks from what they called the Orient then, from Asia, and other kinds of cloths. And so that was Leeuwenhoek's day job. But when, and he was wealthy enough, eventually he could hire other people to do a lot of the work for him, and that gave him some leisure time. And one of the things he did was he made lenses. He made these little beads of glass, is essentially all they were. Very, very smooth, very highly polished, very clear. And when light would strike them, the light rays would be bent in such a way that the image passing through it would be magnified. I'm not a physicist, and I don't really fully understand optics, but in essence, that's what he was doing. And you can see here in his hand is his microscope. All right, it was no bigger than, say, the size of a playing card, really. Just a little square of metal with a lens mounted in it, and then a little pin. I'll show you. It's a little thumb screw thing that had a pin on it, to which you would affix your object you were trying to visualize, and you'd hold this thing up to your eye and look through the lens, all right? And the image that would pass through that lens would be received by your eye, of course, and that would be perceived as being much larger. And he could, with that single lens, excuse me, achieve magnifications of 300x, which means it would appear 300 times larger than it really was, which for a single lens is pretty striking. That's nobody, you know, in his day could come up with anything even approaching that. So he had a special knack for making these lenses, and it was such a prestigious thing for him. He got such a kick out of it. He kept it secret. He really, this sort of violates one of the principles of science, is that, you know, we share, we let other people use our information and what we do, so that, you know, we can have full disclosure of what we've done and make our results public. And, you know, there's lots of quality control that affords. But Leeuwenhoek didn't do that. He kept his lens shaping, his lens sculpting technique to himself. And but, you know, on the flip side of that, he used his microscopes to visualize a lot of things. And what's more, instead of just looking at them and saying, "Oh, that's neat," he recorded what he saw. Right? So he observed very carefully. He took measurements, he made descriptions, and they were very, very thorough. And that's really, when we talk about the scientific method, that's the first step in the method, all right, is to do that. And so he was setting the ground. He was laying the groundwork for people to take those observations and extend them from there to follow through with the scientific method, right? So that's you know, that the importance of that cannot be understated, right? This was an amateur scientist, but he was doing really, really high level work for that time period. Okay? In fact, other scientists, other biologists realized how significant his work was, and they invited him to present his findings at scientific meetings, which are a really big deal. We still have scientific meetings all the time. Different groups of scientists meet every so often to give lectures and presentations about what they're doing. At the time, you know, the people who were doing science typically were hired by royalty, you know, kings and queens and so forth. You know, they would hire these folks to hold positions in, you know, sort of a cabinet, if you will. And they then would have meetings. The Royal Society of London, for example, was a group, it still exists, but they would have regular meetings, and they were some of the people that actually invited Leeuwenhoek to come and speak. And he was the toast of Europe. I mean, he was really, really highly regarded. Here's his microscope. You can see this person's thumb up here that gives you an idea of the scale of this thing. It's a very small thing. And so a specimen would be mounted on the top of that little pin there, and you would hold this up to your eye as this guy down here is doing. And usually there'd be like a candle flame or something behind that to provide illumination to let you see what you were supposed to be looking at. Very crude, but enough to kind of get the ball rolling. So before Leeuwenhoek, people didn't realize that there were microbes. The microbial world was completely unknown. They were affected by these things, but they had no idea of knowing that the Black Death, you know, the plague, was caused by a bacterium. You know, and other kinds of infectious agents that they dealt with. They had no idea. They had no idea that fermentation of milk sugars is by bacteria is what is central to the making of cheese. They had no idea that fermentation was being caused by bacteria to make wine, from grape juice. So this revealed this whole new world that had to be understood. All right, so like I said, Leeuwenhoek with his microscopes was doing observation. He was doing a lot of it, very detailed observations with quantification and the whole works. He would write up his findings. And then, people later could take those observations and think about problems that they would reveal, right? You've got some problem, a burning thing that you don't understand, and you want to try to understand it. So you put forward a hypothesis, which is essentially a temporary explanation for the problem, all right? And in fact, scientists aren't the only people who do this. Scientists are not the only scientists. That's kind of a dumb thing to say, but if you're a homeowner, for example, and you come home and you flip on the light and it doesn't come on, there's a problem, right? You've made an observation. And so what do you start doing? You start thinking of all the possible explanations for why the light didn't come on, right? Well, what do we call those possible explanations? Those are hypotheses. Or say, you're a homeowner and something goes wrong with your plumbing. Your toilet doesn't flush. Or, or your car doesn't start. The point is that in daily life, we apply these same sorts of principles. Scientists are just applying these approaches, this approach to dealing with things in the natural world that demand explanation. So, hypothesis is a temporary explanation for something. Like if you tried to turn on your lights and they didn't come on, you might say, well, the light bulb was burned out. There's hypothesis one. An alternative hypothesis is you didn't pay your light bill. Another alternative hypothesis is somebody hit a light pole, and so the electricity is out on your entire block, or something such as that. Those are hypotheses. So then we take the hypothesis and using a deductive approach, we say, if this explanation is true, then we predict some event Y. So if X is true, then Y should happen. All right. And so that's typically how we do the testing of these hypotheses. So we structure an experiment. All right. Or just more detailed observations to test this if-then proposition. And, we generate data, numbers, measurements, counts, those sorts of things. Say you've got an idea that a particular type of substance will make your tomatoes grow really well. Something you suspect would be an excellent fertilizer. What you could do, you're going to say, if I apply this substance X, then I'm going to see better growth in my tomatoes, right? It's a very Mickey Mouse scenario, but let's say that that's the case. Well, you'd set up an experiment. You might plant, a certain number of plants in a part of your garden that doesn't get the fertilizer, and then a certain number of plants in another part that do get the fertilizer, and then you can compare their growth. But, there's a whole lot more to it than that, because you've got control variables like, if one part of your garden is shaded, and that happens to be where you plant the plants that don't get the fertilizer, then, and they don't grow, it probably wasn't the lack of fertilizer that didn't make them grow, it was the lack of sunlight. So when you do experiments, you have to control all of these other variables. And that, that's true of any scientific investigation. You want, you want consistency. So, like if you have a garden plot, you want to make sure that all of that area gets equal sunlight throughout the day. You want to make sure that the soil has the same quality in all parts of that garden plot. Um, and there's probably other variables that I'm not thinking about. You want to make sure that the same amount of water is applied to the plants in all parts of the garden plot, so that the only thing that could affect them would be the fertilizer. All right. Everything else is controlled, constant, and the fertilizer is varying. That's how you want to do experiments. All right. So you set up an experiment to test your hypothesis. You get numbers, you get data. You look at that data, you analyze it statistically, and then you make a judgment about whether or not the data indicate that the hypothesis is false or valid. All right. Notice I didn't say true. We don't prove things in science. Calling things true, proving things, that's a little bit strong of a statement, because what it says is that idea, if it's supported by the numbers, by the experiment, is going to be unequivocally true for now and forever. And that just doesn't happen. So we say valid or supported for the time being. We reserve some doubt, because it could be that we actually, despite our best efforts, didn't set up the experiment properly. It could be that we didn't analyze the data properly, or something such as that. So we've got to take that into account. Now, if you do reject a hypothesis, if the data show that it is not going to work, yeah, we go ahead and claim that it's false and just toss it out. So that's something. Falsifying hypotheses is easy, and it's something that we don't have qualms about. But saying that something is true, we're going to reserve some doubt about that. So use the word, say that you have validated a hypothesis, or you've verified it, or you've supported it. That makes it seem less, certain. Certainty has no real place here. So yeah, those are the two conclusions. Either reject or support the hypothesis. And if you've tested a hypothesis and you support it, and then somebody else reads your study, that brings up another point. There's a sixth thing that has to be included here. I'm going to say publish, but publicize. When you finish a study, you need to tell the world what you did, and so you write it up and publish that in a journal, and let other scientists look at it, and they'll give their judgment. They'll pass judgment on it. So they might find problems with what you've done, and will critique what you've done, sometimes very, very strenuously. And the end result of this is that it just makes science that much better. Scientists sort of police themselves this way. Without publishing, without making things public, that doesn't happen. So you would finish some work and keep it to yourself, and maybe go from there thinking that you have all the answers, when in fact you've not found out anything. So publishing is vital. You depend on this network of other scientists to kind of reinforce, or sometimes to critique what you've done. So if say you've done a study and you have supported a hypothesis, and somebody else looks at that and says, I'm going to do the same study, I'm going to do the same experiments to see if I can get the same result. And let's say they do that, and they support the hypothesis, and then a third person supports it, and multiple people replicate what you've done, which is also an important thing in science. We have to replicate what we do. If they all say, hey, yeah, it's looking good, it's a pretty strong hypothesis, then instead of calling it a hypothesis, we'll elevate it to a more glorified status, and we'll call it a theory. So in biology, there's not that many theories, because what we're studying is very complex. A lot of people don't get that. They think that chemistry and physics and so forth are really complex. And they are, they're hard. But think about a living thing and being able to generalize about how it functions, and think about all the variables at work and determining how that living thing functions. It's enormous, right? Living things are infinitely more complex than non-living, just physical objects. And so, yeah, biologists are sometimes described as having physics envy, because the physicists and the chemists have all kind of theories, and they even have laws. A law is a really, really strong theory, a really strong hypothesis that, for all intents and purposes, is regarded as fact. We still reserve a little doubt, but you know, it's something that's gotten so much support that nobody in their right mind would dispute it. There are a few of those in biology. We'll talk about one. Well, we'll talk about a couple of them. The most obvious one is the theory of natural selection, the theory of evolution by natural selection. That's the one that has the most support. We also have the biogenesis theory, and the symbiosis theory, and some others that are really, really pretty strong. So there's a few of them out there. But anyway, that's the scientific method. And so, yeah, about the time microbiology was taken off, the scientific method was something that people were really latching onto and accepting. And so they kind of developed in concert with one another. So this is a microbiological. This is pertinent to microbiology because it involves a virus. This is from in the 18th century, work by Edward Jenner, who was a British physician, and one of the major scourges, one of the most horrible diseases ever in the history of humankind is smallpox. Like I say, we've eliminated that, thankfully. It doesn't affect humans anymore, but at one time it killed many, many people. Here in North America, Native Americans succumbed to smallpox epidemics en masse. I mean, whole villages were wiped out by it. It had been brought by Western Europeans. One of the things that Edward Jenner noticed, and of course this was a very concerning thing for him, so he was always kind of thinking about it. One of the things he noticed was that girls who were given the job of milking cows, milkmaids, was what they were called, didn't seem ever to develop smallpox. They were, essentially free of smallpox infection, whereas other segments of the population uniformly seem to be affected by smallpox. The probability of death due to smallpox or being debilitated by it was pretty high in other groups, but not in these milkmaids. So that was curious, and he also observed that if they did get anything like a pox, it was a more mild infection that they got on their hands. And they call this cowpox, right? From handling cows, the udders of cows, they would acquire occasionally some agent that caused the cowpox. Didn't have any idea what it was. Well, that was something that wouldn't be discovered for well over 100 years later. But what Jenner thought to do was to take some of the exudate, that is, some of the fluid, some of the pus in these pustules, these little lesions on the cowpox or the milkmaid's hands, and transfer some of that fluid to a person who had never been challenged with any kind of pox. He thought maybe this cowpox is similar to the smallpox, and maybe exposure to the cowpox affords some sort of immunity to the milkmaids, and maybe that same immunity can be transferred to others. And so that's what he did. Now, by today's standards, what he did next would be considered criminal, right? Incredibly unethical. No one would allow it. It would land you in jail. But at the time, things were different. The social hierarchy was different, and the way people related to one another was different. But anyway, Jenner called a young boy basically off the street and said, "Here, I want to do this little experiment. Maybe this will keep you from getting smallpox." And so he made a little incision in the boy's arm and transferred some of the cowpox exudate from a milkmaid to the boy, and watched what would happen. Then that was followed with introduction of a smallpox sufferer's exudate. Somebody that had smallpox, the real thing, some of their fluid from their blisters would be transferred to this previously treated young person. And lo and behold, what happened was the child didn't get sick, didn't get the smallpox. The earlier treatment with the cowpox exudate seemed to have some protective effect. And he then coined the term vaccination, right? He called this procedure vaccination. And that's the first example of that. And, he went on to replicate his work. I mentioned that was an important aspect of science. If you support a hypothesis one time, that's well and good, but the more support you can give it, the stronger of an idea it is. So he actually did this himself, and he went on to inoculate 23 more subjects with the cowpox fluid, and then challenged them with smallpox fluid, and found that it didn't make them sick. So that's the beginning of vaccination. That theory spread over the next 100 years, and you can see with smallpox anyway, it's been eradicated. In 1979 is when that happened. Anyway, we now have other vaccines, and those are incredibly important as well. Yeah, there's these terms: variolation, inoculation, and immunization. Those are all more or less synonymous terms. Normally today we use vaccination and immunization. All right, so there's that. All right, so I'm going to go ahead and stop it there. They've gone on for quite a while, probably longer than I should have. And we'll go ahead and I'll put up, I think it's one more lecture we'll do it, maybe two, but you'll have plenty of stuff to be reviewing. I will also periodically provide little synopses of what I've covered, and I'll put those up as well. We're going to try to do the best we can. So anyway, good night. I'm gonna stop there and be looking for another lecture here shortly. Okay, and I'll number these. I'll say lecture 1 of 3 on the themes of microbiology, and then lecture 2 of 3, lecture 3 of 3, or however many there are. So that's how we'll do that. All right, I'll be putting them up there on eLearn. All right, so there you go. Thanks.